Hey guys, welcome to your latest installment of your video lectures on genetics, and at this point, very special genetics. In this one, we are going to talk about, as you can see, codominance and multiple alleles. And hopefully they are just as simple for you as all of the other ones have been. I'll try to break it down really simply. So the last special genetics we did was incomplete dominance, and our saying was incomplete is in between. And the reason we said that was, for example, if a gene expressed incomplete dominance, that would mean if you crossed something like a red flower and a white flower, that all of the offspring would be pink flowers. It would be this in-between phenotype. So it would be a blended phenotype. Codominance is similar to that, but it's not going to be exactly the same. So our saying for this is co is both. And what that means is both phenotypes, both physical traits, are actually going to be equally expressed in an organism that inherits the two different versions of the gene, or as we have been calling it, the heterozygote. And we're going to use, again, different letters to represent different alleles, just like we did with incomplete dominance. So instead of like a capital R and a lowercase r, like we did in the regular Punnett squares, we're actually going to use two totally separate letters. And the example is an easy one, so we're going to do chickens. This chicken that you see over in this picture is called an erminette chicken. And as you can tell if you look at the picture, it has this kind of striking coloration. So it has black feathers and white feathers in kind of like a beautiful alternating pattern. And that's because of codominance. So if this chicken, or if a chicken, an erminette chicken, inherited two copies of the black feather gene, then all of its feathers would be black. It would be a totally black chicken. On the other hand, if it inherited two white feather genes, then all of its feathers would be white. But if it inherits one of each, so one parent gives it the black feather gene and one parent gives it the white feather gene, that's when you get this chicken that we have pictured with both black and white feathers. And if we compare this to incomplete dominance, remember, if we had like black and white and we were doing incomplete dominance, then this chicken would be like gray. It would be an ugly gray color. And that's clearly not what we see here. We see both totally black feathers and totally white feathers expressed at the same time in this organism. So let's practice. You're going to cross two black and white chickens. So basically two heterozygous chickens. And there are two questions you're going to need to answer. What is the probability that their offspring will be totally black? And what is the probability that their offspring will be heterozygous? So take a minute and try to set up that Punnett square using your B's and W's, just like we saw on the slide before, and then come back and check the answer. So pause the video so you can practice this Punnett square. Okay, let's check our work. So here are the parents. They are both heterozygous, so a B and a W across the top and a B and a W down the side. And if we did it correctly, this is what we should see as the possible combinations every time these chickens actually have offspring. So what is the probability that their offspring will be black? Well, remember, to be black, that chicken needs to inherit two copies of the black allele. So there is only one box out of these four, or about a 25% chance that their offspring will be black every time they have babies. And then what is the probability their offspring will be heterozygous? Again, meaning they inherited two different versions of the allele. And if we look, we have two heterozygous offspring possibilities. That would be a 50% chance. And then, of course, there would be a 25% chance that that offspring would be white, or that they would have a totally white offspring. So that's codominance. It's very simple. We're going to carry this idea through to the next example that I'm going to give you, but we're going to add something to it. So there's something called multiple alleles, which is actually really common. And as we know, each individual, so you or me, we have 2 parents. So that means we can only have, at max, 2 versions of an allele for every gene because dad gives you one copy of a gene and mom gives you another copy. That's all you get. So you could only have 2 alleles for every gene because those, again, are your 2 homologous chromosomes. So you're 1 through 23 from mom and you're 1 through 23 from dad. But some genes, actually lots of genes, have more than 2 possible alleles. So like in our chickens, right, we had the black allele and the white allele. But for many genes, especially in complex organisms like humans, there's more than just 2 options, like more than just black and white. And that's what multiple alleles means. It means that there's many possible alleles, and then that means that there are many possible phenotypes, at least 3 or more possible phenotypes for an organism for that particular trait. The one that we are going to look at is blood type. So if you have ever given blood, received blood, or just have gone to the doctor, sometimes they actually test you and they tell you your blood type. But your blood type has, on your red blood cells, so your red blood cells carry oxygen all throughout your body. They pick it up at the lungs, and they carry it all throughout your body, dropping off oxygen as they go. But they also have on their surface, embedded in their cell membrane, they have proteins. And depending on the type of blood that you actually have different proteins embedded on the surface of your red blood cells. So here are the different versions of the gene you can get, and they are notated, they're expressed, or written a little bit differently than we've seen before. So these are I's. So I know it looks like just like a lowercase l. That's actually a capital I in this font. So we have a capital I with kind of an exponent A. So we have the A allele. We have our capital I with an exponent B, our B allele. And then we have little I, lowercase I. And the reason we represent it this way is we can't just say A and B because then we're totally losing this allele. We won't have any way to represent it. So the fact that we kind of put these as exponents gives us a little bit more wiggle room in how we represent the alleles. So here are the 3. So we have 3 different versions for this particular gene instead of just 2. That's why we say it's multiple alleles. So if you have the A allele, that means you create what we call the A protein on the surface of your red blood cell. So the A allele is instructions for making the A protein. If you have the B allele, then you make the B protein on the surface of your red blood cells. If you have the I allele, that actually doesn't code for any proteins. So it's recessive. It doesn't actually make any proteins that go on the surface of the red blood cells. And here's a picture for you. Don't worry about the word antigen for right now. Just look at the picture. So you have your 4 different kinds of red blood cells. In blood type A, which is what I am, my red blood cells would have the A protein, which in this case is represented by these little green little pyramids or triangles. Blood type B, their protein is totally different. It's represented by these little blue dots. But what's interesting is when we get to type AB, they actually make both. Like equally, they express A proteins on the surface and B proteins on the surface. So that's actually codominance. So A and B are codominant with each other. They will, if you inherit both of them, you will express both of them together. And blood type O, no proteins on the surface at all. So just to kind of give you a little visual representation of what is actually happening for each of these things. So A and B, just like I just said, are codominant, which means if you inherit a copy of the A allele and a copy of the B allele, you're actually going to express physically both proteins on the surface of your red blood cells. And both of those are completely dominant to the I because I doesn't make anything. So they're both kind of mask that I gene. So here, you're going to have to write this down. So there's a spot on your sheet for you to write this down. So to be blood type A, So I am blood type A. There are two possibilities for my genotype, and you're going to write down both. I can be homozygous, meaning each of my parents gave me the A allele, or I could be heterozygous, meaning one parent gave me the A allele and one parent gave me the I allele. Either way, my red blood cells are going to look like this. They're all going to express type A. For type B blood, it's pretty much the same. So I could be homozygous for B, meaning I got 2 copies of that B allele, or I can be heterozygous for B, which means I inherited 1 copy of the B allele and 1 copy of the recessive I allele. And either way, my blood cells would look like this one over here, this blood type B in the picture. If I am blood type AB, phenotype AB, I only have one possible genotype. There's only one way to get that, and that's if you inherit the A and the B allele. So one parent gives you the A allele and one parent gives you the B allele. And again, your blood type is going to look like this one over here. It's going to have both proteins expressed on the surface equally. And with type O, again, one genotype you could possibly be, because O is recessive, so you need to actually inherit 2 copies of that particular allele in order to be blood type O. So remember, take a moment, write these down. It's at the very bottom of, I think, the back page of your sheet. And you are going to need to know these for when you go through and practice. So let's do a practice problem. So we have a wife with O blood, so her genotype would be little I, and the husband with AB blood, and you should be able to go and check the genotype you just wrote down for AB blood. There's only one. And I want you to figure out what's the possible genotypes of their children. So I'm just going to show you real quick, because people get confused, how you're going to set up the outside of your Punnett square, the parents. So it's going to be like this. Little I is just like, people don't generally have an issue with that. But when you set up these, be careful. IA, that's one thing. That's representing the A allele, and IB is representing the B allele. So we're going to put each of them separately, just like you would like B and W, or big R and little r. So take a minute, set up your Punnett square, do your Punnett square, and then we're going to talk about the possible genotypes of the children. Okay, so here's what you should get. The possible genotypes of the children. Remember, genotype is the actual combination of alleles you inherit from your parents. So it's not asking about blood type. We're not asking what are the possible blood types. So the possible genotypes of the children are IA little I. I have 50% IA little I and 50% IB little I, because that's what both of these are. And what that would mean if they asked you about phenotype is it would be 50% type A and a 50% chance of type B blood. Hopefully easy enough. And then for the bottom question, you might not have to do a Punnett square. So if you need to, you can take a minute to do one. So let's say dad has type A blood and mom has type B blood. They have a child, at least one child, and that child has type O blood. So based on that information, what are the parents' genotypes? If you need to take a moment to pause and do the Punnett square out, pause and do the Punnett square out. All right, I'm not going to show you the Punnett square for this one. So if you have questions about it, you're going to have to ask when you come back in. But here's the answer. These are the parents' genotypes, IA little I and IB little I. Because each parent, my dad who's type A and the mom who is type B, they must carry that recessive allele in order to give it to their child. Because that child, his or her genotype is little I, little I. So they must have inherited that one copy from each of their parents, which means that both parents have to be heterozygous for A and heterozygous for B. Because if they weren't, if they were homozygous, IA, or IB, then there'd be no way that their child could actually be type O for blood type. So I hope that was simple. And now you have some problems that you're going to need to practice. And if you have any questions, make sure that you have them ready to ask us when we return from break. Thanks so much, guys.